Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So today we are going to discuss cancer biology, in which we were discussing about hallmarks of cancer. Today we will be discussing about the Warburg effect. Warburg effect. So the deregulating cellular energetics, it is quite an important hallmark of cancer. And normal cellular metabolism, which is quite important, and it is a fundamental process which enables energy production, the biosynthesis pathway through the breakdown of micronutrients like glucose. It is quite important because that is going to provide energy for the survival. So glucose, it is metabolized via oxidative phosphorylation, anaerobic glycolysis, or aerobic glycolysis. By this mechanism, it produces energy in the mitochondria or in the cytosol. So depending on what condition is there, like whether the cell is living in aerobic conditions or anaerobic conditions, what pathway glucose is going to follow for ATP production is decided. But what happens is that this choice of following the pathway instead of following the oxidative phosphorylation pathway, the tumor cells, they follow the aerobic glycolysis pathway, means they convert the glucose to lactate even in the presence of oxygen. So this deregulation of cellular energetics is seen in the cells, and that is known as Warburg effect. So Warburg effect was first described by Otto Warburg in 1920. So this was described by Otto Warburg in 1920. So this is a phenomena where the cancer cells and the uncontrollably growing cells, they make the choice to utilize the aerobic glycolysis instead of oxidative phosphorylation. So using aerobic glycolysis instead of phosphorylation, oxidative phosphorylation that happens in mitochondria. This choice is done by cancer cells. So this is seen in cancer cells. So they preferentially utilize aerobic glycolysis. Aerobic glycolysis means generally in the absence of in the absence of oxygen, glucose is converted into lactic acid. But the conversion of glucose in lactic acid in the presence of oxygen is known as aerobic glycolysis. So this is opted by the cancer cells. So the question is, why do cancer cells opt this pathway? Like, why do not they go for oxidative phosphorylation? Because oxidative phosphorylation will be producing much more energy compared to the aerobic glycolysis. So different proposals were made to predict that why do those cells, why do the cancer cells, they opt for the aerobic glycolysis. We'll discuss about that proposals as well. So let's first summarize what happens in the glucose metabolism pathway. So glucose follows few major metabolic pathways. First, it can undergo glycolysis. Okay, in this, the glucose is converted into the pyruvic acid. So this generates some substrate level ATP and the NADH. Okay, so now the NADH generated in this cycle, they enters into the tricarboxylic acid cycle, the TCA cycle, as well as the pyruvate, which is generated in the pyruvic acid, which is generated in the glycolysis. Both of them, they enter into TCA cycle, the tricarboxylic acid cycle, the Krebs cycle, which produces CO, which produces CO2 by oxidizing the pyruvic acid, and it also produces NADH as well as FADH2. Okay, so the NADH and FADH2, which are produced in this cycle, as well as the NADH, sorry, not this NADH. This NADH doesn't enter in TCA well. The pyruvate enters into the TCA. So NADH and FADH2 produced in the this TCA cycle, and NADH produced in glycolysis. All of them, they enter into the electron transport chain reaction which happens in the mitochondria to to do the ATP synthesis. So this produces ATP. So let me draw it in a better way. So what happens is that glucose is converted into pyruvic acid, okay, plus some NADH and ATP. So this pyruvate enters into TCA cycle to produce some NADH, FADH2. Okay, so this NADH that is produced from the glycolysis pathway as well as the TCA pathway 1, 2, and 3, all of them they enter into electron transport chain reaction to give out the ATP. So the ATP generation through this electron transport chain reaction is much higher than compared to the glycolysis pathway. Okay, then this happens in the aerobic condition in the presence of oxygen. But when present the oxygen is absent, when there is no oxygen, the glucose is converted into lactic acid, the lactate. Okay, this is what we call as anaerobic glycolysis. So in this process, the energy generation is less compared to what is done in oxidative phosphorylation. So this is what we call as oxidative phosphorylation. So then the question was, why do the tumor cells they opt for this pathway? Why do they do the lactate production in the anaerobic condition? Even though the cells have oxygen, so tumors they are growing in the normal conditions, right? So they already have oxygen, so why do they opt for it? Why do they produce lactate by the help of lactate dehydrogenase? So the here the enzyme which works is lactate dehydrogenase. So the different proposals were given for that. So let's list down the proposals. The proposal one states that the aerobic glycolysis provides the ATP at faster rate compared to the oxidative phosphorylation. So it says that the ATP synthesis is rapid compared to what we see at the in the oxidative phosphorylation. Even though oxidative phosphorylation allows cell to generate much more ATP, but the rapid ATP production is seen in the aerobic glycolysis pathway. So this anaerobic glycolysis which happens in the absence of oxygen, this is followed in the cancer cells and it is known as aerobic glycolysis. So the cancer cells they show the lactate formation even in the presence of oxygen. And the reason behind this is that maybe the aerobic glycolysis it does the rapid ATP synthesis. So this proposal was made. But then the question is, why are the ATP demands not limiting for proliferation, and why do alternative mechanisms, what kind of alternative mechanisms for rapid ATP synthesis are there in the cancer cells, which that is which makes the tumor cell to to make the ATP demand at the second priority. So this is a question which arises with this proposal. The other proposal says that whatever intermediates are produced in the glycolysis, those they are first. So the cancer cell they first follow glycolysis pathway. Okay, so whatever glycolysis produces, the intermediates are produced in that biosynthetic pathway. They are used for other biosynthetic pathways like the pentose phosphate pathway, which produces ribose 5-phosphate for nucleotide biosynthesis. Then amino acid synthesis can be done using the glutamine and serine, using the precursors for glutamine and doing the serine biosynthesis. Then lipid biosynthesis can be done from the acetyl CoA, which is the intermediate of the glycolysis as well. So using those intermediates, you can use the bios, you can use the you can use those intermediates in other biosynthetic pathways. So this enables rapid cell division. And thus allowing cells to grow uncontrollably. So now you have the nucleotide. So the tumor cells have their own machinery to synthesize nucleotides, amino acids, and lipids, and they have all of the raw material. So the raw material providing the supply of raw material will be done by using the intermediates of glycolysis. Okay, so this proposal also explains why the other pathway, why the cellular metabolics they are deregulated in cancer cells. Then other proposal says that the Warburg effect enhances immune evasion. Okay, so it enhances immune evasion. So this enhances tumor microenvironment. Okay, so this favors cancer progression. So lactate accumulation that leads to the acidification of the tumor microenvironment. So what happens is that the immune cell activation will be suppressed, which will lead to T cell dysfunction, macrophage polarization. Okay, and that will also promote. So the lactate accumulation will promote first the T immune cell suppression. So immune cell activation will be suppressed. Immune cell activation will be suppressed. Okay, then it also promotes angiogenesis. We'll learn about angiogenesis in next lecture. Okay, then it also enhances tumor invasion and metastasis. When we learn about those hallmarks, we'll learn about how this exactly happens. But for now, just remember that the proposal says that the tumor microenvironment is modified, and the Warburg effect enhances the immune evasion. Okay, so this is the fourth proposal says that Warburg effect, so deregulation of cellular metabolics is done so that the cellular signaling is supported, and this modulates the reactive oxygen species. So what happens is that the Warburg effect, it enhances cellular signaling. Okay, and once cellular signaling is enhanced, the cells are able to regulate the pathways in a better way, and as well as it modulates the reactive oxygen species, ROS. So ROS signaling, the moderate level, it moderates the proliferation. So whenever excessive ROS is there, it leads to the oxidative stress. Okay, so moderating the ROS, the reactive oxygen species, this helps the cancer cells to proliferate uncontrollably. Then there will be epigenetic regulation of the which will influence the gene expression, like acetyl CoA from glycolysis. It will do the histone acetylation, and now this will help in better cellular signaling, so that the proliferation will be uncontrollable. The specificity in signaling is quite unclear. What happens in that part, it is not really well laid by researchers, and we don't know what would metabolites level be influenced by the metabolic flux. Like what happens if certain metabolites increase, and how do they affect the levels of other metabolites. So metabolic flux is somewhat, it is unknown. So there's a question whether the cancer is a metabolic disease or not, whether it is a metabolic disorder or not, and a lot of study is going on in that field right now. So there are multiple regulators, the oncogenic regulators, that has effect on Warburg effect. For example, MYC, it upregulates the glycolytic enzymes such as HK2, LDHA, so they promote the glutamine metabolism. Then there is the HIF1 alpha. Okay, so this HIF1 alpha, this induces expression of glycolytic gene under hypoxia conditions, means whenever the oxygen level is low, and even under the normoxia, it induces the expression of the glycolytic genes. Then there is ATK mTOR pathway, which regulation of which enhances the glucose uptake via GLUT1 transporter, and this stimulates glycolysis. Then there is P53 pathway, the tumor suppressor pathway. This suppresses glycolysis and promotes oxidative phosphorylation. So these multiple regulators of these multiple oncogenic regulators, as well as the tumor suppressor pathway, they helps in the metabolic reprogramming, and this is how the cellular energetics, it is deregulated in the cancer, so that it will support the uncontrollably growing cells, because those cells need a lot of nutrients, they need a lot of oxygen, so you need to change the metabolism. And that is why in the cells, the over the oxidative phosphorylation, other pathways are preferred, and that is what we call as the Warburg effect. The aerobic glycolysis means conversion of glucose into lactate under the presence of oxygen. So this is seen in muscle cells as well. So whenever there is hypoxic condition, muscle cells convert glucose into lactic acid, which get accumulated in our muscles. So this is seen in the cancer cells as well. So with this, we are done with the Warburg effect. In next lecture, we will learn about other hallmarks such as angiogenesis, the suppression of immune responses by our by the tumor cells. We'll also learn about the invasion and metastatic properties of cancer cells. Till then, see you. Bye bye.